The topic dealt here is somatic embryogenesis. What is somatic embryogenesis? Process by which bipolar structures resembling a normal zygotic embryo is developed from somatic cells. These cells are independent and do not have vascular connection with the rest of the tissues. Another term that we come across is embryoids. Embryoids are small, well-organized structures similar to sexual embryos which are produced by means of tissue culture. They are capable of giving rise to the whole organism. They have the plumule and the radicle. The process of somatic embryogenesis leads to the production of somatic embryos. The characteristics of a somatic embryo are that they have single-celled origin and bipolar in nature. That is, a single cell is capable of giving rise to the shoot and the root meristem. These are independent cells and have no vascular connection with that of the explant. They are anatomically and physiologically comparable to the normal zygotic embryos. Based on the origin of somatic embryos, somatic embryogenesis is divided into two types: direct somatic embryogenesis and indirect somatic embryogenesis. In direct somatic embryogenesis, the embryo formation occurs directly from the explant, from pro-embryonic determined cells. There is no callus formation. As you see in the figure in your extreme left. Here, callus formation occurs from the tip of the leaf explant. In case a callus is formed from the explant and after that embryoids developed, then it is known as indirect somatoembryogenesis. The figure in your extreme right shows the formation of callus from the explant, which is then triggered to form small embryoids. The regeneration of somatic embryos is a multi-step process. It can be divided as: first, formation of pro-embryogenic cells or masses; second, somatic embryo formation; third, maturation; fourth, desiccation process; and lastly, plant regeneration. Desiccation tolerance is one of the most important property of seed acquired during seed development, basically late seed development. It enables the seeds to survive during storage and environmental stress. Desiccation of somatic embryos leads to dehydration of embryos to varying extents and improves its efficiency in germination. In some cases, these embryos are partially dried in high humidity. Desiccation is believed to break the dormancy of the somatic embryos and also promotes accumulation of nutrients like proteins which are utilized during germination. The whole process of this somatic embryogenesis can be divided into two distinct stages: phase 0, which is embryo initiation, and second, embryo production. In embryo initiation, differentiated somatic cells acquire embryogenic competency and proliferate as embryonic cells. These require high concentrations of auxins as stimuli. The second, embryo production, where these embryonic cells are capable of differentiating into somatic embryos. At this stage, very low levels of auxin or no auxin is required. In phase 0 of somatic embryogenesis, competent cells are formed. These cells are the cells that have reached a transitional state and have already started to become embryogenic, but they still need a stimuli, external stimuli by means of growth hormones like auxins. In the next stage, the cells are said to be embryogenic cells. That is, these cells have finished with the transition stage from a somatic cell to a stage of embryogenesis, and they require no further external exogenous applied stimuli such as growth hormones. These embryonic cells resemble meristematic cells, however, they are generally smaller, more isodimetric in shape, with densely stained nuclei and have a denser cytoplasm when compared to the normal meristematic cells. The different stages of growth in somatic embryogenesis of a dicot plant can be summarized as: firstly, a single cell is de-differentiated in an appropriate nutrient media composition and multiplies. Second, induction or initiation or acquisition of embryogenic competency by these cells. This could be a direct or an indirect method. This is then followed by the developmental stages and the maturation stages of the embryo, which are globular stage, the heart-shaped embryo, and the torpedo stage. These torpedo stages then develop into a plantlet. Figures here illustrate normal embryogenesis and somatic embryogenesis. The figure on the left shows normal embryogenesis in plants. Here, the male gamete fuses with the egg cells in the embryonic sac to form the zygote. This is then followed by series of cell division from 2-cell stage to 16-cell stage, followed by globular shape, heart-shaped, and torpedo stage, and lastly to the young embryo, which has the shoot apical meristem and the root meristem, which gives rise to the shoot and the root respectively. In case of somatic embryogenesis, a somatic cell is triggered by external stimuli to become embryogenic cell, and these embryogenic cells divide to form the 2-cell stage, the octat stage, followed by the globular, the heart-shaped, and the torpedo stage. This is followed by the cotyledon stage, where the shoot and the apical meristems are destined. The figure here shows indirect somatic embryogenesis. Here, various stages of development of somatic embryos can be seen, like the globular stage, the heart-shaped, the torpedo stage, and the dicot embryo. The figure here shows the sequential stages of indirect somatic embryogenesis from a style or stigma explant. Figure ABC shows the formation of callus. Figure D shows the presence of globular somatic embryos. Figure F shows the somatic embryos showing cotyledonary leaves, and G shows the plantlet showing a leafy shoot and the root, which can be regenerated into a complete plant. The figure here shows the histological pattern of somatic embryogenesis. Figure A shows the cells which are single-celled, diad, triad, and in tetrad stages. Figure B shows the globular to the early heart-shaped stage. C shows the quaternary embryo. D shows a later quaternary embryo stage. The technique of somatic embryogenesis has been successful in monocots, dicots, and conifers. However, there are slight variations seen in the different stages of somatic embryo development. Like in monocot, the globular stage is followed by the scutellar and the coleoptral stage. In dicots, we have already studied, it follows the proembryonic, followed by the globular, the heart-shaped, torpedo-shaped, and the quadrilaterary or the bipolar embryo-shaped. In conifers, it follows the globular stage, the early quaternary stage, and late quaternary stage. The figure below shows the difference between direct and indirect somatic embryogenesis in both the dicots as well as the monocots. Thank you.